Hello, my name is Savannah Gann, and I conducted research on starch and deflocculation, the effects of Calgon on ancient starch recovery, and the archaeology of food laboratory with Dr. Crystal Dozier at the Department of Anthropology at Wichita State University. Ancient starch research, the field for which I conducted the following experiment, is the area of archaeology concerned with extracting, identifying, and analyzing starch residues in an archaeological context. While starch has had a long history of study in other fields, ancient starch analysis as an archaeological marker developed rapidly only in the late 19th century. Since that time, ancient starch analysis has been applied to numerous studies, including those focused on stone tool function, the emergence of agriculture, the domestication of plants, the evolution of human diets, cultural mobility and movement, as well as land use patterns and vegetation history. These starch-focused research topics and more help us to understand past civilizations and cultures in ways that were previously inaccessible. However, due to the relative newness of the field, modifications are still being made to the methods and standards used within ancient starch research, specifically through experimental studies and the testing of archaeological practices. When artifacts or sediment samples are recovered from archaeological sites for the purpose of starch or pollen analysis, it is extremely important that the granules be extracted without any alterations or damages, as these starch cannot be replicated. While there are various techniques that may be used to process materials recovered from the field, one of the most common extraction techniques utilized by ancient starch researchers is deflocculation. Deflocculation is the process in which sediments containing starch residues are soaked in a strongly charged ion solution to release the microbotanical remains from the clay particle aggregates. And this just means that a sediment sample recovered from a site or an artifact must be soaked in a solution which separates the microbotanical remains, like the pollen or starch that is being analyzed, from the clay and silt from which the sample was extracted. For this particular study, we used Calgon, or sodium hexametaphosphate, a rapid and inexpensive solution commonly used in the deflocculation process. However, there are various conflicting conclusions about the use of Calgon as a deflocculant, which we hope to address. To better understand the effects of Calgon on starch granules, we conducted the following experiment to determine if the amount of time that starch is left in a deflocculant solution, in this case Calgon, affects starch recovery. To conduct this experiment, we first prepared the starch solutions. We began by dissolving 8 lycopodium tablets and a 10% hydrochloric acid solution to use as the control for the study. By centrifuging the lycopodium and hydrochloric acid mixture at 3,300 RPM for 3 minutes, we were able to concentrate the lycopodium spores at the bottom of the test tube. We then decanted or poured out the excess liquid and vortexed the sample to ensure that we obtained as many of the spores as possible. We continued by measuring out 0.2 grams of common cornstarch with 100 milliliters of a 5% weight by volume Calgon solution. We then mixed the lycopodium with the starch Calgon solution and added 5 milliliters of the mixture to 7 separate test tubes, signaling the beginning of the timed soaking periods. The tubes were then centrifuged at their respective timestamps. In this case, the rinse, 5-minute, 10-minute, 30-minute, 1 hour, 2 hour, and 24 hour, and then decanted and vortexed. Due to time constraints and dense starch distribution, only the rinse, 1 hour, and 24-hour samples were included in this study. Slides were prepared with a 1-to-1 ratio of the sample solution and glycerin, then counted manually with the human eye and a mechanical counter on a microscope. Results from the experiment suggest that starch recovery can decrease by over 50% when soaked in Calgon for a 24-hour period. The table shown here lists the starch and lycopodium quantities for each slide, along with their respective starch-lycopodium ratios. Figure 1 at the top shows the starch-lycopodium ratio from the rinse sample in 10x magnification, while the figure just below that shows the same sample in polarized light. Looking again at the figures, you can see that the white granules in the top are the starch granules and the yellow granules are the lycopodium tracers. In this experiment, the amount of time in which starch is left to soak in Calgon affected starch recovery. Results from the experiment show that starch sample sizes may decrease by over half when left in Calgon for 24 hours, an amount of time common for deflocculation in the field of archaeology. The significant decrease in starch recovery from the rinse to 1 hour samples suggests that ancient starch researchers, hoping to extract as many granules from their source material as possible, should soak Calgon for less than 1 hour. The continual decrease from the 1 hour to the 24-hour further suggests that prior deflocculation with Calgon for the standard 24-hour period has harmed starch recovery in previous experiments and studies. Starch density in this experiment was also incredibly high, with granules unevenly dispersed across slides and a human-based counting system used. As such, granule abundance and human error may have impacted data collection. To combat these issues and to further understand the effects of Calgon on starch recovery, additional studies should be conducted with more controlled measures of starch distribution and a more efficient data collection method. Further research should also observe other starch taxa, such as potato or wheat starches, to test varied reactions of starch to Calgon. Deflocculation with Calgon has previously proven to be an inexpensive and efficient way of cleaning specimens recovered from archaeological sites. However, these experiment results suggest that soaking artifacts or sediment samples in Calgon for 1 hour to 24 hours can significantly reduce starch recovery, implying potential reduction of starch quantity in previous ancient starch research. To better understand the effects of Calgon on starch recovery and the consequences of timed soaking periods, further data will be collected from soaking samples under the 1 hour mark, such as the 5-minute, 10-minute, and 30-minute samples. Listed here are my references. Thank you, and I will now take questions. So starch recovery and analysis is a relatively new field in archaeology, but we can actually learn a lot. So if we're able to extract the ancient starch for which I conducted this experimental study, if we're able to extract those starch and analyze those and determine maybe what particular type of starch is present in a potsherd or a sediment sample, we can learn about foodways, ancient foodways, such as how people were preparing food, how long they were storing it, maybe cultural and physical mobility, where they were moving to and from in order to gather these types of food. So starch research can actually tell us a lot about ancient lifeways, food. It's a way of bringing women back into the narrative because a lot of the time women were the one preparing the food. And these aspects of culture may have been inaccessible prior to starch research because we didn't know what types of food they were preparing or where they were gathering their food from. I think that the current methods of starch recovery have been in question, just not to an extent to this degree. I have read literature that has tested other deflocculant solutions, such as just plain water or a different solution. Calgon has been used since the late 70s as a deflocculant solution, and it has been questioned, but not to this extent. I think the relative newness of the field, considering archaeology is such an established field and ancient starch research is a new subject within that, I think that it has been in question, but not to this degree. And clearly there's more research that needs to be done, which is why I will be continuing this research under the 1 hour mark.